the ghost DNA of West Africa. Imagine a phantom breathing in your lungs, a biological entity that pulses through your veins but possesses no fossil record, no skeletal remains, and no place in the official history of the Earth. This is not a thought experiment. It is the current reality of human genomics. You have been taught that the human family tree is a well-mapped lineage, a clear progression from a single African source. However, modern computational biology has just detected a massive, invisible occupant in our genetic house. The mainstream doctrine suggests that modern humans emerged approximately 300,000 years ago and occasionally interbred with known cousins like Neanderthals or Denisovans. It is a clean, safe narrative of a unified species. But in 2020, researchers at UCLA, using sophisticated deep learning algorithms to sequence the genomes of West African populations, hit a wall of stratigraphic impossibility. They discovered that up to 19% of the genetic ancestry in these populations does not come from modern humans, nor does it come from Neanderthals or Denisovans. It comes from a source that does not exist in the archaeological record. This is what scientists call ghost DNA. The thermal signature of this genetic introgression suggests a divergence from the human line nearly 625,000 years ago. To put that in perspective, this mysterious ancestor was walking the Earth, evolving, and mastering its environment for hundreds of thousands of years before the first modern human was even a biological possibility. Yet, there is an absolute archival silence regarding their physical form. Where are the bones? Where are the tools? The data indicates a molecular fusion occurred around 43,000 years ago, a massive interbreeding event that fundamentally altered the human blueprint in West Africa. The investigative paradox is staggering. How can a species contribute nearly a fifth of the genetic makeup of millions of living people and yet remain entirely invisible to the shovel? Mainstream academia refuses to acknowledge the implications of a super-archaic lineage that was likely more adapted to the harsh tropical environments than we were. We are looking at a population that survived the catastrophic resets of the Pleistocene, only to be absorbed into our own line. The chemical analysis of these ghost segments reveals genes linked to tumor suppression and hormone regulation. This was not a random mutation. It was a sophisticated biological acquisition. We are not a pure species. We are a chimeric construct, carrying the code of a shadow lineage that dominated the continent for 500,000 years before disappearing into our own blood. If these beings were advanced enough to survive the climate shifts that killed off other hominids, why is their existence relegated to the footnotes of genomic studies? The technical weight of this discovery shatters the out-of-Africa simplicity. It suggests that Africa was not a cradle of a single species, but a melting pot of multiple, highly distinct human lineages that lived and died in a world we can no longer see. We are walking around with the operating system of a ghost. How many other silent partners are hiding in our code, waiting for the right algorithm to reveal their presence? The victim of this global crime scene is the concept of human purity. We are the survivors of a forgotten war for genetic dominance. Who was the entity that gave us 19% of our identity, and why has history been scrubbed of their name? The red-haired giants of Lovelock Cave, Nevada. A heavy, suffocating scent of ancient decay fills the air as you descend into a lightless cavern in the Nevada desert. In 1911, two guano miners, looking for fertilizer, broke through a sealed entrance to Lovelock Cave and uncovered a scene that should not exist in North American history. Among the thousands of artifacts were sandals measuring 15 inches in length, equivalent to a modern size 20, and mummified remains that stood over 7 feet tall. The most jarring detail, however, was the hair. A deep, vibrant, rusted crimson. The mainstream doctrine insists that the first inhabitants of the Americas were small-statured hunters who crossed the Bering Land Bridge. Any mention of giants is dismissed as a misinterpretation of skeletal remains or a result of chemical staining from the cave soil. They tell us that the red hair is simply the result of nitrates in the guano leaching the pigment out of black hair over centuries. But this safe story collapses under the weight of forensic reality. The investigation into the Lovelock remains reveals a structural density in the bones that far exceeds the limits of standard human physiology. These were not modern humans with a growth disorder. They were a distinct biological lineage built for extreme physical exertion. Chemical analysis of the red hair has shown that the pigment is not a topical stain but is embedded deep within the medulla of the hair shaft, a clear genetic marker of a non-indigenous population. Furthermore, the artifacts found alongside them, intricately woven duck decoys and complex textiles, suggest a level of technological sophistication that contradicts the image of a savage outlier. Local oral histories from the Paiute people speak of the Ctach, a race of red-haired giants who were in a state of perpetual warfare with the native tribes. These were not gods, but a competing human lineage. The Paiutes claim they eventually trapped the giants in Lovelock Cave and set a massive fire at the entrance, suffocating them. When the cave was excavated in 1911, a distinct layer of charcoal and scorched stone confirmed this exact event. Yet, the Smithsonian Institution reportedly took possession of the primary skeletal evidence, and since then, there has been a systematic archival silence. The paradox is clear. How does a 7-foot-tall, red-haired population establish a presence in the Great Basin thousands of years before European contact? If they were merely a coincidence of nature, why were the skeletons removed from public view and the research suppressed? The official timeline of human migration cannot account for a Caucasian-featured, giant-statured lineage in the American West. We are looking at a suppressed chapter of a global conflict between human subspecies. The thermal scarring on the cave walls tells the story of a final, desperate stand. These beings were not just tall. They were a specialized branch of humanity that has been methodically erased from the record to preserve the Bering Strait narrative. If the sandals were size 20 and the bones were real, then the history of America is not a story of discovery, but a story of replacement. Who were these red-haired giants, and what other impossible occupants were here before us? The Solutrean hypothesis, the first Americans. A razor-sharp flint blade lies on the deck of a scalloping boat in the Chesapeake Bay, pulled from the seabed alongside the tusks of a mastodon. The blade is made of a high-quality rhyolite, bifacially thinned with a precision that defies the capabilities of the Clovis culture. It is an exact technological match for the Solutrean tools of Ice Age France and Spain, separated by 3,000 miles of Atlantic ice and 10,000 years of official history. This single artifact, the Sinmar blade, is a structural tear in the veil of American archaeology. The mainstream doctrine is a rigid fortress known as Clovis First. It states that the first humans entered America 13,000 years ago via a land bridge from Siberia. According to this narrative, no one crossed the Atlantic until the Vikings, and certainly not during the height of the last glacial maximum. The idea of Ice Age Europeans navigating the North Atlantic in skin-covered boats is treated as a maritime impossibility. However, the evidence from the Sinmar blade is undeniable. Carbon dating of the mastodon remains found with the tool placed it at 22,000 years old, nearly 10,000 years before the Bering Land Bridge was even open. This is a stratigraphic impossibility for the mainstream timeline. Furthermore, genetic markers found in certain Native American populations, specifically haplogroup X, show a lineage that originated in the Near East and Europe, not East Asia. This DNA does not exist in Siberia. It is a molecular signature of a cross-Atlantic migration that occurred while the world was encased in ice. The investigation by researchers like Dennis Stanford and Bruce Bradley suggests a sea ice edge migration. These ancient mariners would have followed the edge of the Atlantic ice pack, hunting seals and seabirds, effectively using the ice as a highway from Europe to the east coast of America. This is not a theory. It is a logistical reality based on the toolkits found at sites like Cactus Hill and Meadowcroft Rock Shelter. These sites contain pre-Clovis layers with Solutrean-style technology, yet they are often ignored or dismissed by the academic establishment to protect the safe Siberian story. The paradox is intellectually destabilizing. If the first Americans were, in part, from the Solutrean culture of Europe, it collapses the entire political and historical framework of the New World. It suggests a global, interconnected Ice Age civilization that was capable of transoceanic travel at a time when we are told humans were barely surviving in caves. The suppression of the Solutrean hypothesis is a masterclass in archival silence. Why is the Sinmar blade not in every textbook? Why is the haplogroup X data treated as an anomaly rather than a lead? We are looking at a forgotten peak of human navigation followed by a catastrophic reset that wiped the memory of the Atlantic crossing from our collective consciousness. The history of the Americas is a crime scene where the evidence of the first arrivals has been buried under a layer of academic dogma. How many other first arrivals have been erased to keep the timeline linear? The blue-blooded gods. In the high-altitude reaches of the Andes and the deep archives of ancient Sumer, there are persistent accounts of a ruling lineage with blue blood. While modern culture treats this as a metaphor for nobility, biological anomalies suggest a far more jarring reality. There exists a potential human lineage whose circulatory system was not based on iron-rich hemoglobin, but on copper-based hemocyanin. When hemocyanin is deoxygenated, it is clear. When it carries oxygen, it turns a vivid sapphire blue. The mainstream doctrine tells us that hemoglobin is the only viable method for oxygen transport in large mammals. Any deviation is dismissed as a medical deformity or a mythological fabrication. They claim that blue blood is merely a reference to the prominent veins of pale-skinned aristocrats who didn't work in the sun. This is the safe explanation designed to keep our biological history within the narrow confines of the iron-based evolution. The tear in the veil comes from the technical efficiency of copper-based blood. Hemocyanin is actually more efficient than hemoglobin in cold, low-pressure environments. For a species evolving in high-altitude mountain ranges or under intense atmospheric pressure, copper-based blood isn't a mutation, it's a superior adaptation. There are documented medical cases of methemoglobinemia, where blood appears chocolate brown or blue, but the blue-blooded gods narrative refers to a stable, hereditary lineage. Investigation into the royal Grail lineages of Europe and the dragon courts of the East reveals a bizarre obsession with maintaining blood purity, often to the point of extreme inbreeding. Could this have been an attempt to preserve a recessive, copper-based biological trait? Ancient texts describe these rulers as having a cold disposition and a different physical glow. Thermal signatures of copper-based life would be significantly different from our own, as copper is a superior conductor of electricity and heat. The secret detail lies in the chemistry of the gods. Many ancient cultures insisted their rulers were descended from the stars, or came from the high places. If a branch of humanity developed in an environment with low oxygen and high copper availability, their entire molecular fusion would be distinct. We see high copper concentrations in the Oligocene fossils, but mainstream science refuses to look for it in human remains. The archival silence regarding the chemical analysis of royal mummies is deafening. Before you delve into the next 4 forbidden lineages, consider this: who benefits from your ignorance? Join our inner circle by subscribing now to unlock the archives they once stayed closed. Is the blue blood of our history a forgotten biological reality? Were we once ruled by a human subspecies whose very chemistry was designed for a world we no longer inhabit? The question isn't whether they existed, but whether their descendants are still among us, hidden by the very blood we consider normal. Denisovan supergenes. You are standing at 15,000 feet on the Tibetan Plateau. The air is thin, lethal to most, yet the people here move with a grace and stamina that defies human physiology. They aren't gasping for air because their blood contains a genetic superswitch called EPAS1. This isn't the result of millions of years of slow, Darwinian adaptation. This gene appeared in the Tibetan genome almost overnight, and it didn't come from us. It is a 100% match for the DNA of the Denisovans, a human lineage that officially went extinct 50,000 years ago. The mainstream doctrine suggests that humans are the pinnacle of a slow, grueling climb toward peak performance. We are told that we are the winners of the evolutionary race because of our superior intellect. But the EPAS1 gene proves that we didn't win, we downloaded. Our survival in the most extreme environments on Earth is due to interbreeding with a species that mainstream science barely recognized until a single finger bone was found in a Siberian cave in 2008. The investigation into the Denisova cave and the Baixia Karst cave in China has revealed a shocking truth. The Denisovans were not primitive cousins. They were a high-altitude, high-tech branch of humanity. In the same soil layers as their remains, archaeologists found a green chloridolite bracelet, polished to a mirror finish using a high-speed drill that modern toolmakers struggle to replicate without diamond-tipped equipment. This is a technical weight that academia cannot carry. How does an extinct species possess drilling technology 50,000 years ago that we only mastered in the industrial age. The paradox of the Denisovan supergenes is that they are still active. They aren't just in Tibet. They are in the Melanesians of the Pacific and the Aboriginal Australians. These populations carry up to 6% Denisovan DNA, including genes for immune system, overdrives, and skin pigmentation that protects against extreme UV radiation. This wasn't a coincidence. It was a strategic molecular fusion. We are looking at a lineage that was specifically adapted to extreme Earth, the high peaks and the deep islands, environments that modern humans were physically incapable of colonizing on their own. Why was the Denisovan lineage erased from the official timeline until 2008? The archival silence for over a century of archaeology in Asia is a global crime scene. We have found their tools, we have found their jewelry, and we carry their supergenes. Yet we are told they were a failed branch of the tree. The EPAS1 gene is a smoking gun. It proves that the modern human is a scavenger of traits, a composite creature that only survived by absorbing the superior biological tech of the Denisovans. What else did we download from these forgotten architects of the high places? If they had high-speed drills and genes for super survival, why are we told they disappeared? The reality is that the Denisovans never truly left. They are functioning within your cells right now, providing the code that allows us to inhabit a planet we weren't originally built for. We are the inheritors of a ghost civilization, the forbidden graves of the Darim Basin. In the middle of the Taklamakan Desert, the desert of no return, lies a graveyard of the impossible. In the early 20th century, explorers began finding mummies preserved by the salt and dry air of the Darim Basin. The beauty of Lulon and the Sherchin man look as if they died yesterday. They have auburn hair, high-bridged noses, and are dressed in intricate wool tunics. There is only one problem. They are 4,000 years old, and they are Western Eurasians in the heart of ancient China. The mainstream doctrine dictates that the East and West were isolated until the Silk Road was established around 130 BC. The idea of a Caucasian civilization flourishing in Western China 2 millennia before that is a dangerous concept for the official historical narrative of both the East and the West. For decades, the Chinese government suppressed the findings, and Western academics ignored the data, hoping it would remain buried in the sand. This is a classic case of archival silence. The tear in the veil comes from the textiles. Chemical and structural analysis of the Sherchin man's clothing revealed a tartan weave identical to the Celtic Hallstatt culture of Central Europe. The wool came from sheep that were not native to Asia. This isn't just a physical presence. It is a technological signature of a European culture that migrated thousands of miles through hostile terrain to establish an outpost in the desert. DNA testing has since confirmed that these people carried West Eurasian maternal and paternal lineages. They were not travelers. They were a settled, thriving population. The investigation into the small river cemetery, Xiaohe, revealed even stranger details. The graves were marked by massive wooden poles that looked like oars, and the bodies were buried in boat-shaped coffins, covered in cowhide. In a desert where water is a death sentence, they were buried as mariners. The paradox is staggering. Why would a Caucasian, boat-building, tartan-wearing culture establish itself in the most landlocked, arid place on Earth? The technical weight of the evidence points to a catastrophic displacement or a highly organized, lost colonization effort. These people were tall. Some of the men were over 6 feet, and they possessed advanced metallurgical knowledge. Yet, the official history of China makes no mention of this forbidden lineage. They are treated as an anomaly, a coincidence of the desert air. But 1,000 years of Caucasian presence in the Darim Basin is not a coincidence. It is a suppressed chapter of human geography. Why were these people there? Who were they running from? Or what were they searching for? The Darim mummies are a silent witness to a time when the world was far more mobile and ethnically complex than we are allowed to believe. The Great Wall of history isn't made of stone. It's made of the secrets we refuse to translate. These auburn-haired ghosts are a reminder that the map of the ancient world has been redrawn to exclude those who don't fit the modern narrative. The hobbit species of Flores. In 2003, on the remote Indonesian island of Flores, a team of researchers looking for evidence of human migration stumbled upon a biological impossibility in the Liangbua cave. They found the skeleton of a 30-year-old female, barely 3 feet tall, with a brain the size of a grapefruit. She wasn't a child. She was a fully grown adult of a completely unknown human species, Homo floresiensis. But the chilling detail wasn't her size, it was the date. She lived only 12,000 years ago. The mainstream doctrine has always maintained that Homo sapiens became the sole human inhabitants of the planet after the Neanderthals vanished 30,000 to 40,000 years ago. We are the lonely species. The discovery of the hobbits shattered this. It meant that while our ancestors were painting the walls of Lascaux and beginning the agricultural revolution, a second, tiny human species was hunting giant rats and pygmy elephants just a few islands away. The investigation into the Liangbua cave revealed a paradox of intelligence. Homo floresiensis had a brain volume of only 380 cubic centimeters, smaller than a chimpanzee's. Yet they produced sophisticated stone tools, controlled fire, and organized communal hunts of the stegodon. This is a molecular impossibility, according to the mainstream brain-to-body ratio theory of intelligence. How does a creature with a primitive brain display modern behavioral complexity? The official narrative has no answer, so it attempts to label the find as a diseased modern human with microcephaly. This safe explanation was debunked by the structural analysis of the wrist bones and the feet. The hobbit had a trapezoid bone in its wrist that is identical to an ape's, and feet that were disproportionately long, requiring a high-stepping gait. This was not a deformed modern human. It was a relic lineage that had evolved in isolation for nearly 1 million years. The technical weight of the skeletal data proves a long-term, successful human branch that survived right up to the dawn of modern civilization. The secret detail lies in the local oral tradition. The people of Flores tell stories of the Abu-Gogo, small, hairy, forest-dwelling people who stole food and spoke in a murmuring tongue. These stories aren't treated as myths by the locals. They are treated as historical encounters. Some accounts suggest the Abu-Gogo were seen as recently as the 19th century. If they survived the volcanic eruption that supposedly wiped them out 12,000 years ago, are we certain they are truly gone? The archival silence regarding the potential survival of Homo floresiensis is profound. If a parallel human species lived alongside us until the Holocene, it means our dominance was never guaranteed. It suggests that the world was once a place of multiple, competing human realities. The hobbits are a tear in the veil of our self-importance. We were not the only ones to make it to the finish line of the Ice Age. We were just the ones who stayed to write the history books. The O-negative mystery. Look at your blood. If you are O-negative, you are a biological anomaly. You lack the rhesus Rh factor, a protein found on the surface of red blood cells in 85% of the human population. This protein is named after the rhesus macaque because it is a universal trait among primates. Every other ape, monkey, and modern human ancestor possesses this factor, but O-negative humans do not. In the eyes of evolutionary biology, you are a stratigraphic impossibility. The mainstream doctrine tells us that Rh-negative blood is simply a random mutation that occurred somewhere in the Basque region of Europe or the Atlas Mountains of Africa. It is a safe story that ignores the most violent biological paradox in nature: hemolytic disease of the newborn. When an Rh-negative mother carries an Rh-positive fetus, her body does not recognize the child as its own. It identifies the fetus as an invading pathogen, a virus or a parasite, and produces antibodies to attack and destroy the baby's blood. Nature does not design a species to reject its own offspring. This does not happen anywhere else in the animal kingdom. This is not natural selection. It is a molecular fusion error. It suggests that the Rh-negative lineage and the Rh-positive lineage did not evolve from the same source. If we all came from the same primitive primates, why does the O-negative mother's body treat the standard human blood as a foreign threat? The investigation into the O-negative lineage leads to the Cro-Magnon expansion and the mysterious Basque people, who have the highest concentration of Rh-negative blood in the world. Their language, Euskera, is a language isolate. It has no connection to any other tongue on Earth. It is a linguistic and biological fortress. Historical records show these populations were physically distinct, with larger cranial capacities and different bone densities. They were a peak population that appeared suddenly in Europe, replacing the Neanderthals with a technology and biology that seemed to come from nowhere. The archival silence regarding the origin of the Rh-negative factor is chilling. There is no ancestral Rh-negative primate. There is no evolutionary precursor. It appears in the human record as a finished, functional, and highly distinct blood type that is fundamentally incompatible with the rest of the species. Is it a forbidden lineage that was introduced to the gene pool? Was it the result of a catastrophic reset that forced two different human branches to merge? The question of O-negative blood is the ultimate unanswered question of human history. If you are Rh-negative, you are carrying a biological marker that says you do not belong to the same lineage as the rest of the world's primates. You are a living artifact of an unknown origin point. If we didn't evolve from the same ancestors, then the story of human life is not a tree, it is a laboratory. Who were the original negative donors, and why is their existence the most suppressed secret in biology?